Hi, I'm Eric. Thank you everyone for giving up coffee and playing with all the Nanopore products to come listen. As Miles said, my work focuses on early warning. And so really what we're trying to do is get ahead of infectious diseases at high-risk interfaces. So we need to be able to anticipate, to prevent, to detect, and to respond to infectious diseases. However, early warning in and of itself requires active surveillance, and that active surveillance is really costly and time-consuming for any of you that have ever done it. It's also not often done in the easiest of places. So in Cambodia, we are out in places like lakes and rivers and markets and things like that. So in terms of avian influenza, while avian influenza or human influenza pretty much disappeared during the pandemic, avian influenza has been going up, okay? And that's important not only because, we're detecting it here in Asia. I'm sorry, I'm having a lot of technical difficulties today. But also because it's been going into new places. So into Europe, into South America, now there's issues with potentially going into Antarctica, and there have been a number of human infections with these clades as well. And there's probably a lot more that are undetected or unreported. The other issue is that it is also spilling over into numerous mammalian species, things like these seals in Chile. So this is a major issue not only for pandemic risk, but also for biodiversity. And the question really becomes, it's not if the next influenza pandemic will occur, it's when. Okay, so statistically, there will be another influenza pandemic, likely within the next 7 to 10 years. And so it's critical that we get ahead of those things. And working at the Institut Pasteur in Cambodia, we're a non-for-profit research institute of public utility since 1953. We've been at the animal-human interface in the region for over 25 years, okay? I'm not going to go too far into that, but we're using sequencing in a number of different capacities to look at different diseases, some of which I've listed here within the Virology Unit. In terms of Cambodia, for those of you that don't know, Cambodia is a very small country in the Indochina Peninsula. It is a hotspot of emerging and endemic infectious diseases, and there's a very high reliance on agriculture, which means that avian influenza is highly prevalent here. So anytime I go out to a market or anything like that, about 20 to 40% of birds or 30 to 50% of ducks will be positive for some form of avian influenza. And so we've been doing active longitudinal surveillance in these markets for over 10 years. We see a lot of patterns in the seasonal spread. And a lot of times these are associated with festival periods where people are actually producing more birds and purchasing more birds. It's also critical, since now we're moving into more environmental samples. So environmental samples, as some of you do wash water and wastewater, things like that. We're also doing environmental sampling to increase speed, comprehensiveness, biosafety for ourselves and the other people, but also animal welfare, and of course cost. And so when we're in these markets, and here's a picture of a bird market in Phnom Penh, we've been looking at a number of different kinds of subtypes of environmental samples, but lately we've been focused on air. And we've been taking air samples in a number of different markets. We can now do this in a number of different ways. So we can do it handheld, where we can get up and close with the birds. We can get up and close with the defeathering machines. And we can even actually do this remotely now. So we do things like strap these to drones and fly them into bat caves and things like that. But also we have what I call terrestrial drones, which are really just remote-controlled cars that we've strapped these things to. And we do this in markets, okay? So we can drive these around markets and get a better view of what's going on in that market itself. Why we're doing this and why we're, and we're also doing this in the wild, obviously, with any kind of outbreak that we might have as well. So why I'm telling you all this is that we need to be able to do genomics from these samples directly, okay? And so what we can do with those samples is obviously what we all know is not only detection, but also outbreak tracing and tracing things in markets, but also mutation analysis, vaccine and drug discovery, as well as animal and public health. So, obviously, rapid full genome sequencing, both in the lab and in the field, is absolutely critical to what we do, especially for detection and response times. We've been using what we call integrated index multi-segment PCR for avian influenza virus. This is first developed by our colleague Peter Thielen at Johns Hopkins Applied Physics Laboratory, but we've been using it quite extensively. What we can do with this barcoding is actually reduce our response times down from, say, 33 hours, which would be with Illumina, down to sample to sequence in about 7 hours on a MinION in our lab. Okay, and this has been critical for a number of reasons. It's critical not only for monitoring subtypes of concern that we know of in these markets, but also for discovering and assessing new subtypes. It works on all subtypes of influenza A. So here we've discovered some new H14 viruses in ducks in Cambodia. It's also been critical to responding to human cases in Cambodia, which we've had 2 H9 cases in the past 2 years, which we used this quite successfully. And then this year, we had some cases of H5, which we were able to go from sample to sequence in our laboratory and get that data out for public health response within 24 hours. Okay, we got the sequence within 7, but we got it to the authorities within 24, and it was up on GISAID within 3 days of actually having that sample in our hand, and that's critical for response time. However, we all know that it's not always easy to do sequencing. There's known problems with avian influenza and influenza sequencing on ONT, and things like data quality, gene segment coverage, things that can affect our response times because we're not confident in our data. So with the new R10 chemistry, we were hopeful that this would actually improve our quality. So I'm going to tell you a little bit about the work that we're doing, asking can the R10 chemistry improve our rapid AIV sequencing. And so what I'm going to just show you here is that we've taken about 45 samples of avian influenza virus that we've taken from an environment and birds, compared R9, R10, and Sanger sequencing to actually look at these things. And what we see is that R10 has higher data output, read quality, and read lengths compared to R9, which is a great advancement that we've seen. I'm not going to go through all of these slides because I don't have a lot of time, but I'm happy to talk about the details later. In addition, R10 has lower insertion and deletion rates, and this is very, very clear, especially in low complexity regions. So if you look at deletions and insertions, you see that the R9 has much, much more than the blue R10. Okay, I think that's very clear to see. And on a functional level, if we look at something like the multibasic cleavage site of these highly pathogenic viruses, which is critical for avian influenza itself, so for those of you that don't know a multibasic cleavage site, normally avian influenza is only able to infect the trachea and the cloaca of a bird. Having a multibasic cleavage site, which is it's a very low complex region in the cleavage site, it allows it to bind to multiple ubiquitous proteases throughout the bird, and it can go systemic, causing major outbreaks of poultry death. On a functional level, R10 was able to resolve that low complexity region in 90% of our samples, compared to only 60% of the same ones that we ran on R9. So you can just see, just from these 3 points, that this quickly improves our sequencing. It enables us to do these things faster and cheaper, okay? And it also allows us to have more reliability and speed. So we have increased speed, we have decreased cost, and we've increased accuracy. So we're more confident in what we're doing. There's still a lot of work to do, and I'm happy to talk about this in question and answer and things like that. But I think the main point here is that every second counts between detection and response. Okay, so any advancement that we can get, especially like what we're getting with the new R10s, is critical for what we're doing. And I think that's very clear from the past that in different pandemics in history, this has been a critical point. Okay, so if I just pull out some examples, things like HIV, SARS-1, Ebola, and even the 1918 pandemic, earlier detection and earlier response would have saved millions and millions of lives. So just the ability to do this very quickly with higher confidence allows us to respond much, much faster. And with that, I will just say thank you to all of my partners, and of course, thank you to Nanopore as well.